Hi, everyone. My name is Alisa Omelchenko, and I am from the University of Pittsburgh, but I'm also in a joint program at CMU in the CPCB Computational Biology program. So I simplified my title. I promise you're in the correct room. And today I will be talking about how we use networks and systems approaches to uncover TFH differentiation core program. And so TFH differentiation is incredibly important across the board in many biological concepts, from infections to autoimmunity. But this process is quite complicated, and because of its complexity and all the different signals that you're getting to get these TFH cells, there have been a few inconsistencies across experiments and species. For example, we can look at TGF-beta or activin A, which has been shown to be really important for human in vitro TFH differentiation, but not in mouse. And even when we look at the same species, we can look at IL-12. And so some papers will say that IL-12 promotes TFH in infection, and the paper here on the right is
Okay. All right.
great. So what we have is we developed a few data sets that are bulk. We have some ATAC-seq, cut and run with 3 different markers, and gene expression with the help of our collaborators, Vinay and Shiv. And we separated that data based on 4 TFH differentiation states. And this is not really the part that's at question because those are really well known. But with these 4 different separated bulk states, we can start to take a look at what is happening in that trajectory. And we take these data sets and we can put them through networks like a gene regulatory network. And we can start combining the gene expression with the epigenomic signals and start to identify which transcription factors really drive the changes across these different states. So we can take an ATAC-seq data set and create a network, and we can weight that network with the gene expression and use personalized PageRank to identify those driver transcription factors that are different across the 4 different states. And we can take the top genes that are ranked across all 4 of these different epigenomic signals and further consolidate them down to 152 driver transcription factors. Now, there's a lot of overlap from those initial 400 genes to those 152, but there's also a few that are different across those 4 different data sets. And this also allows us to capture some things that maybe one of these data sets would have missed. First, when we take a look at that rank, which is differential across those 2 different states, so more highly expressed in GC versus early extra GC state. And if we look at the gene differentiation or gene differential, we see that they're not fully correlated. So here in blue, I'm showing the genes that we picked out. These are our 152 genes, and they are not very highly differentially. And so if you're just using differential expression, a lot of these genes would be normally missed. And if we take a look specifically at those genes, we can use that gene rank to start to separate them across which ones are important for your early extra GC state and which ones are important only for GC TFH. And a lot of them fall under the fact that they're important for both of the states. And specifically, we have BCL6 and PRDM1 or BLIMP1, which are famously known to control GC-TFH differentiation. And PRDM1 here is not very highly changed across these two states, so often it would be missed. And here the coloring is green if it's expressed highly in early extra GC state, and purple if it's expressed higher in the GC TFH state. And I'll be using that coloring throughout the rest of the talk. So I guess the take-home message here is that regulation is not always determined by that gene expression. There are other factors at work where networks can help find those things. And differentiation is not just controlled through transcription factors and gene regulation directly. You also might have signaling cascades and phosphorylation events where these have these physical interactions that are super important. So we also wanted to look at the protein-protein interaction network and integrate these signals through that. So we took the 4 different data sets, we created a gene-centric peak proximity score, and we used that to weight the nodes in the protein-protein interaction network. And what this network propagation allows us to do is to identify subnetworks of proteins that are important for each other. And so if you have some dropout in signal, if all of your proteins around are highly, have high scores, then you can start to find the things that would have dropped out. And also because we permute the networks and we change the edges to a lot of random networks, we do 500 permutations, we can find things that are only true in that biological topography and not true in a random network. So really home in on that biology. This is just a conceptual schematic of the fact that the networks that we find are incredibly specific. So this is showing less than 1% of the whole interactome, and the modules that we find, which are 27, are really sparse and very specific to our task. And I also wanted to mention they are very stable because when we redo this multiple times, we get the same exact modules. And one of the reasons why this is really, really important is that when you want to create hypotheses, it's really important for it to be interpretable so we can go back and say, why are we getting these modules? And start to answer those questions of mathematically, why are they coming up? Where is the signal coming from? And the genes here don't fully matter. I'm just showing like these are the genes that come up. But we get a lot of those known genes that are PD1 and CD200, which are physical interactors in TFH biology. And we also can start doing that same state specificity where here we have an IL-17 module that is expressed higher in the early extra GC, and IL-17 has to be turned off so that you can get these GC-TFH and not a different helper subset. So now that we have our gene sets, we wanted to see how much can we capture of known TFH biology. So we used 2 different papers. We have the Carolella Vinuesa follicular helper cell paper, as well as a paper by Terry Loffer and Andrew Hart from 2020. And so these papers really capture a lot of TFH biology over the decades, right? So they are kind of all of the things that we have found so far, and we want to see how much of that do we capture. First of all, if we just use those raw scores, just that peak proximity score or differential expression, we really get a weak signal, if any signal at all. But when you take those scores and you propagate them through a network, that's when you start to get a lot more signal and a lot more information. So we get about 60 to 70% of that known TFH biology captured with these methods. But the most important thing is that we start to get context. So if we take that Vinuesa paper and we overlap it with the protein-protein interaction network, as well as the genes that we have found, here I'm showing circles are things that are in the paper and we find. Squares are things that we don't find and are in the paper, and diamonds are things that we find that are not in the paper. We start to see how those components that are in these reviews are connected. And if we just use differential expression, we see that it's a lot more fragmented. And you actually lose quite a few of those connections, like IRF4 connection to STAT3 and STAT1 through these interactors. And so that context is really nice to have. And we think that if we found the true core TFH program, then it doesn't really matter what tissue type you have or what data modality you have. What matters is that we found that core program. So we put that to the test, and we use single cell data sets as well as spatial, and we identify a TFH population and a non-TFH population, and then see whether we can separate them based on the gene sets that we see with gene set enrichment. And the first data set that we used came from Susan Ribeiro's lab at Emory, where we used a gut sample from people living with HIV. And again, so now we're changing the data modality, we're changing the tissue type, and we're adding a disease context. And we identified the TFH cells as well as central memory. These are our non-TFH cells. And what we see is that our prioritized gene sets are actually okay at separating the two. A Cliffs delta here, so we use effect size and a p-value, and a Cliffs delta between 0.1 to 0.3 is moderately effective because of how much cell heterogeneity we have. And so compared to the publications, if we just use those publications to try to separate those different populations out, we do better. So our set is just maybe a little bit more generalizable than these published data sets. We also repeated this with the spatial information, and since there's a lack of time, I'm not going to go over that, but we do get such a pretty good separation between TFH and the non-TFH population, and we also can start to see some of that spatial information being incorporated. Further, we wanted to see, well, if we can do different tissue types and different data modalities, can we do different species from this human-derived analysis? And so we took the genes that we had, and we did the same thing with the literature. We took a literature set from Shane Crotty, 2020 for the murine. And then we also have a data set from Amanda Paholick's lab, which did an allergy model in murine with house dust mite. First of all, we have pretty much the same amount of overlap with the murine network as we do with the human network. So that was really interesting to see. And when we look at that single cell allergy lymph node murine tissue, compared to a murine curated gene set, we do pretty similarly at separating those two different cell types. So here we use TFH and resting T cells. And if we use the human data set and human reference set, we actually do 1.5 to 2-fold better at being able to separate these populations. And what this suggests is that, if we look at these populations and again, try to see how are these things all connected, we can color our network by which review paper it came from. So we can take the two human papers that we used, Vinuesa and Hart, and then if it's in that paper, it's colored in orange, and we can use the mouse set. Here it's colored in blue, and the things that are red show up in both of those contexts. And we have this, well-known common network. That's your BCL6, that's your PRDM1, that's your PD1. But there's a lot of things here that are coming from either one or the other side that if you look at the protein-protein interaction network, they're actually very well interconnected. So perhaps this suggests that there's a lot more interconnectivity between the human and the mouse network for TFH differentiation than previously reported. And I think that's really a nice thing to follow up on. And so now that we have looked at our gene sets, we wanted to see, well, what novel things can we look at and what novel things can we discover through the pathways? So we did a pathway enrichment analysis where we took our gene set and looked at different types of databases like Hallmark and PID. And so we have here coming from the bulk data, and then this one is coming from the single cell data, and they're pretty much in accordance of what pathways come up. Interestingly, we see TNF alpha signaling and that it needs to be turned down in the TFH genes in order to get TFH. And this is actually quite a new discovery. This is the only paper that we know of in 2020 that actually showed that TNF alpha decreases TFH. And we also can start to look at the PID network. And here we found this IL-12, IL-27, IL-23 family that also needs to be turned down. And so if you remember at the beginning of my talk, I said that IL-12 is really highly contested in the publication. So in both mouse and human, there's a lot of questions of whether IL-12 promotes or inhibits GC-TFH. So when we go back to our networks and start to visualize the network, this is the PID network for IL-12. We see that IL-12 actually needs to be turned down and that it is highly expressed in the early extra GC. And this is it coming up in one of our modules. And so with the help of our collaborators, Rebecca Elsner and Mark Schlomchik, we did an in vivo murine model to see how that, if we block IL-12, what actually happens in the case of infection, and whether it affects our GC-TFH population. And so we have a heat-killed control here, which means that there's no infection, and that's the normal number of TFH cells. In the case of infection, the GC-TFH cells go down because they turn into Th1 cells to fight off the infection. And what we see is when we block IL-12, you get that phenotype back and you get the same number of TFH cells as you would in the non-infection case. And that is very time specific because when we do, when we block IL-12 later on, what we see is that this is not, we cannot get those GC-TFH back. So what we did is we identified a core TFH differentiation network that's conserved across species, tissues, disease contexts with these network approaches, and that we were able to recapitulate 10 years of publications with just this one analysis, and that it creates a foundation for a lot of functional investigation. We provided some context to the disagreement of IL-12's role in TFH differentiation, and hopefully I did convince you that network-based approaches, I'm sure this audience, I don't have to convince you too much, are a very powerful way to look at these types of things in immunology. I want to say thank you to my lab, specifically Yija and Tracy, my PI who is actually giving a talk at another part of the conference today, Jishnu Das, all of our collaborators that made this possible, and our funding sources. And also thank you so much for having me here today. This was very great opportunity, and I'll take any questions.
We are a few minutes behind, but I think there is time for one question. Are there any questions in the audience? No? Okay, please. Yes.
All right. I just want, maybe I missed it, but can you clarify with the network propagation thing? Because you mentioned you run it and it's the same. I was wondering, is it because the way we propagate is deterministic and therefore it doesn't matter how many times we run?
Well, so it matters because of the permuted networks. So you permute the networks differently every single time. And so that kind of adds a little bit of that. But it does converge on the same group of gene sets. And that's a mathematical, it just mathematically will always converge.
Okay, thank you very much.